Hello everyone, my name is Gemma Geisler, and my chosen BioBit topic is on how horseshoe crab blood is used as medicine and is important in scientific medical studies today. In this presentation, some of the main points I will be covering are why horseshoe crab blood is used, how horseshoe crab blood is used, and some controversies about using horseshoe crab blood as medicine. These two pictures right here show the blood of the horseshoe crab. The picture to the left shows the vein placement of the horseshoe crab, and the picture to the right shows the actual color of the horseshoe crab blood, which is blue, in a glass jar, which I thought was cool visually. Here are some different pictures of horseshoe crabs, and in the middle is a picture of a baby horseshoe crab, which you can see how small they are compared to a human's hand. And then the picture to the far right shows how great and big they can be. The pictures over to the left show how they kind of look, as well as the bottom left, which shows the underneath with all their legs and tail, which some people may find disturbing if you have never picked one up yourself. So, some facts on horseshoe crabs. Their scientific name is Limulus amoebaceae, and they have been around since even before the dinosaurs were alive. There are actually horseshoe crab harvesters out there, and in a study done by the Atlantic States Marine Fisheries Commission, about 100,000 to 150,000 horseshoe crabs are brought in to get their blood extracted yearly, with 464,482 crabs brought in to biomedical facilities in 2018. Physically, they are quite ugly and scary looking to some, but they are actually really cool, special, and unique creatures. Horseshoe crab blood does not look like normal blood, as previously talked about. In fact, it is actually a milky blue color, and it is that way because it contains a very minute amount of copper in it, and it's often described to look alien-like. Who was behind discovering the use of horseshoe crab blood anyway? Well, in the 1960s, Jack Levin and Frederick Bang, who are based out of Woods Hole, Massachusetts, discovered that the horseshoe crab blood had bacteria-fighting properties to it. This was discovered by injecting the horseshoe crabs with bacteria at different levels to see how toxic it could get. And from this, they figured out that the horseshoe crab blood actually clotted around the bacteria, therefore detecting the presence of the danger. Frederick Levin then went on to create LAL, which stands for Limulus amoebaceae lysate. This specifically is the extraction of a protein in the blood cells of the horseshoe crab, and this is what alone detects bacterial endotoxins and produces the clotting, which occurs almost instantly. Now, this picture on the bottom shows the actual extraction process from the horseshoe crabs, and you can see their blood being extracted at different levels and the vibrance of how blue it really is. So, the Smithsonian published an article on how the extraction process actually works. So, when the horseshoe crab is captured, about 30% of its blood is extracted, and then that blood is used for Limulus amoebaceae lysate in a lab. Then, subsequently, the crabs are returned back into the ocean. How is it used today? Now, you might be asking, well, what does it detect bacterial endotoxins in, and how is it used today? Now, in the present day, we use Limulus amoebaceae lysate to detect bacterial endotoxins in vaccines, various different drugs, medical supplies, and medical administered fluids using the same method of injecting the limulus amoebicate lysate into the desired sample and seeing if clotting occurs, in turn saving millions of lives and ensuring safety. Something I found very interesting in my research is that for every COVID-19 shot vaccine administered, a horseshoe crab was used to ensure its safety. The COVID-19 vaccine was created rather quickly under the time constraints given, and a major part of how this was possible was because of the limulus amoebicate lysate testing done to it. Now, some of the controversies about using horseshoe crabs is not knowing exactly how many of these crabs die in the process, but estimates show it can range from a few percent to about 30% or more. However, studies done by the International Union for Conservation of Nature have also shown that the horseshoe crab population was labeled vulnerable in 2016, but since has increased to where they are no longer labeled vulnerable. In conclusion, this overall is a groundbreaking discovery that horseshoe crab blood can be used like this in the first place. Finding the use of limulus amoebicate lysate changed the way modern medicine is approached and how we can verify that the use of vaccines and different drugs is safe for humans. Having the peace of mind we won't get deathly sick because we have horseshoe crabs protecting us. And this is my works cited.